Hi, my name is Zoe. I'm a student researcher at the Vivian Lui lab in the Georgia Cancer Center, and I'm actually next year heading to MCG to get my MD. And I'm here to explain to you guys the importance of PDCs in precision medicine and what our grant is focusing on currently. So cancer is one of the leading causes of death in the US currently. It stands right behind heart disease as the highest killer in the US. And unfortunately, there's not too many super effective treatments for cancer. You might be familiar with chemotherapy and radiation as treatments, and those treatments tend to have a lot of side effects. And the reason this happens is because these treatments actually kill all the cells in that area, regardless of if they are cancerous or healthy cells. The good news is, as technology has progressed and our understanding of cancer has progressed, a new field of cancer treatments has opened up, and these treatments are known as precision medicine, which is what our lab focuses on. And essentially what you do is you take a tumor, you sequence the genome, and you try and find mutations in the genome, and then you try and find specific drugs that target those mutations. And because cancer cells have more mutations than normal cells, the normal cells should not be affected by these drugs. And so what you have is essentially a treatment that does not cause all these side effects and hopefully treats it faster. So the reason precision medicine is so important is because it has been shown in some cases to be more effective than traditional chemotherapy or immunotherapy or radiation, and very promising results have been shown in the literature. But because it's a very new field, we need to do a lot more research before it becomes as common as chemotherapy. And so one important component of this research is the use of PDCs, or patient-derived cultures. Basically, you take a patient's tumor, which is essentially just a mass of cancerous cells, and you process them until they can grow by themselves in a flask, like this one. You can then run a genetic analysis on these cells and use them for all sorts of precision medicine research. Additionally, different populations of people tend to have more common mutations amongst themselves. So a treatment developed from a PDC derived from a Caucasian patient might be less likely to work or not be as effective on a South Asian patient, for example. This makes it critically important to gain samples from different ethnic and racial groups so that these treatments can be developed evenly for all ethnicities and races across the board. Unfortunately, however, medicine is full of disparities in research, and currently no head and neck cancer PDCs that derive from African Americans exist. Our grant is focused on us developing around 5 to 8 of these PDCs so that they can be used for drug testing. The goal is to develop these PDCs and characterize the genome, and then publish the results and share these resources with the rest of the research community to help bridge some of the inequalities in medicine that exist today. So this research was made possible by a UR2 grant obtained from Augusta University by the Vivian Lui lab. Grants like these are so important because they fund research that allows us to develop more treatments that will eventually help patients and help bridge some of the gaps in the inequalities of medicine that exist today.